This video describes the key benefits of the Biolaminin 521 culture system for maintenance and lineage-specific differentiation of human pluripotent stem cells. Biolaminin 521 is a human recombinant laminin cell culture substrate produced and distributed by Biolamina. Biolaminin 521 is a feeder-free, defined and animal component-free substrate for culture of human embryonic stem cells and induced pluripotent stem cells, mesenchymal stem cells, most anchorage-dependent progenitors, and for differentiation and maintenance of specialized cells such as hepatocytes, cardiomyocytes, and neurons. For more controlled and standardized experiments, we recommend passaging pluripotent stem cells as single cells or as small aggregates. These phase contrast images show human embryonic stem cells seeded as single cells or as small aggregates on Biolaminin 521. Within 60 minutes post-seeding, the majority of human pluripotent stem cells seeded on the biolaminin substrates have attached and flattened down, and the cells are evenly distributed across the well. The cells migrate and start dividing, and the day after seeding, the cells have formed small colonies. The cells rapidly proliferate without sharp borders between different colonies and form a homogeneous monolayer that has equal access to the medium and matrix. Contrary, human pluripotent stem cells cultured in thick multilayer colonies often require manual, arbitrary removal of spontaneously differentiated parts. It is to be expected that cell morphology will look different on Biolaminin 521 compared to cells cultured as colonies on feeder or on other feeder-free matrices. After attachment, the cells flatten out on the laminin substrate and will look bigger compared to colony-cultured cells. When the cells grow more confluent, the cells will become smaller, more similar to cells cultured as colonies, with a high nuclear-to-cytoplasm ratio and prominent nucleoli. This is a time-lapse video of human embryonic stem cells seeded on Biolaminin 521. Due to the natural support from the matrix, cells show high motility, survival and proliferation rate. Biolaminin 521 is available as both research grade and cell therapy grade, supporting scientists throughout their cell therapy process, from discovery all the way to clinical applications. The defined and xeno-free Biolaminin 521 culture system is more flexible, easier to use and less laborious compared to a feeder-free based culture system. Biolaminin 521 is a natural substrate for human pluripotent stem cells since laminin is produced and expressed around the cells in the inner cell mass. Via interaction with key cellular receptors, the biologically relevant cell matrix triggers natural cell signaling pathways, leading to more consistent cellular responses and improved cell functionality. Biolaminin 521 culture leads to reduced variation between different human pluripotent stem cell lines, stabilising the pluripotency expression pattern. This, together with a minimal lot-to-lot variability, gives robust, reproducible and reliable protocols. Biolaminin 521 works well in combination with most commercial media brands, and the protocol can easily be made totally defined and xeno-free with your choice of culture medium and dissociation reagent. While colony passaging leads to varying split ratios and passaging intervals, Biolaminin 521 supports single-cell passaging, making it user-insensitive, less laborious and more standardised. These are crystal violet stainings of human embryonic stem cells 24 hours after plating in single-cell suspensions onto a EHS matrix extract and Biolaminin 521, without and with the addition of ROCK inhibitor. Biolaminin 521 supports single-cell passaging without the need for artificial apoptosis inhibitors such as ROCK inhibitor. The split ratio can be adjusted empirically depending on your need. The Biolaminin 521 matrix supports cell survival at low seeding density, and the cells can be cultured to high confluence. Biolaminin 521 also supports clonal culture applications, such as induced pluripotent stem cell reprogramming, gene editing applications, and human embryonic stem cell derivation. Human embryonic stem cell lines can even be derived from a single blastomere, thereby circumventing the ethical issues associated. Human pluripotent stem cells cultured on Biolaminin 521 grow in a homogeneous monolayer without spontaneously differentiated areas. These immunocytochemistry stainings for pluripotency marker OCT4 in pink show a homogeneous positive result for cells cultured on Biolaminin 521. Unlike colony passaging on feeders and other feeder-free matrices, cells cultured on biolaminin 521 can be cultured to near confluence without signs of spontaneous differentiation, and you do not have to spend time cleaning up your culture by removing differentiated areas. These bright field images show a homogeneous cell population on biolaminin 521 compared to a heterogeneous cell population on another feeder-free substrate. Cells grow fast on biolaminin 521 with a stable proliferation rate and maintained genetic integrity. The proliferation rate facilitates the use of low-passage human pluripotent stem cells for production of clinically relevant cell numbers of differentiated cell types. Biolaminin 521 also allows a weekend-free feeding regime. This, together with a minimised variability and the protocol flexibility, adds value and further reduces labour and costs. Besides well-defined and high-quality stem cell cultures, successful stem cell-based therapy requires differentiation methods with sufficient yield and purity. The more authentic culture environment primes the pluripotent stem cells for more efficient differentiation, enhanced cell maturation, polarisation, and organisation of specialised cell types, such as hepatocytes, RPE cells, skeletal muscle cells, and different neural cells. The effect of laminins on the advances in cell-based research and therapy have been published in many high-impact scientific journals. For more information on how to culture human pluripotent stem cells on biolaminin 521 and other procedures, please visit our website at www.biolamina.com.